and digestion. So what I'm giving you right now today is not on tomorrow's lab practical. So I'm going to talk at you for maybe half an hour here, and then I'll let you have the rest of the time to review or whatever you want to do. I put a few updates on mastering. So the pre-lab for blood is now up there, and I set it due for Thursday since you've got a test tomorrow. I think it's really fair maybe to do tonight. Even though we're going to start that material today, that pre-lab's not due until Thursday. And then I think I set lymphatics due for next Monday. So we've got 2 new pre-labs up there. Make sure you check that. And I put the blood and lymphatics quizzes up as well. But of course, there's no penalty for doing those later. They're up and available. Okay? Make sure you check those. And I'll assign exam 1, which will be in 2 parts. One will be over cardiology, and one will be over peripheral circulation. So you'll take those as 2 separate assignments in mastering, but it'll only count as 1 grade. Okay, it'll show up in mastering as 2, but when I put it into your grades in Blackboard, I'll take those 2. So introducing blood.
When did you say that would be available? You didn't say like what?
Oh, when they make it, I have not made it. It will be available later today, probably by like 5 or 6
The
days.
exams?
Hmm?
The exams?
Oh, how many attempts? No, just 1.
No, I mean like what day will it be available? Like,
Today.
Okay.
Yeah.
What?
Yeah. All right, so let's introduce this whole blood thing. We've talked a lot about the circulatory system now. We've talked about peripheral circulatory structures, the heart. We haven't really talked about what's being pumped around in all this, which is blood. So blood is a connective tissue, which means it's got fairly few cells and a lot of matrix. Blood is the only, well, it's not the only, but it's the most prevalent liquid connective tissue. So its matrix is liquid. These are just some slides from your book. If you take a whole blood draw, what's called a peripheral blood draw, and you put it in a little test tube, and you put this test tube in a benchtop centrifuge and spin it at about 10,000 RPM, it'll separate, it'll fractionate into a couple of different layers. That topmost layer is called the plasma. That's the liquid portion of blood. So the matrix of blood is plasma. And it contains proteins, salts, and water. That's what plasma is made of. Protein, salts, and water. Beneath that is the packed cellular component down here. This is called the hematocrit. The hematocrit is composed almost entirely of red blood cells. Okay? So it fractionates typically 55% plasma, 45% hematocrit. There's a little layer between the hematocrit and the plasma called the buffy coat, and that's where most of the white blood cells reside. So that buffy coat there, that contains mostly white blood cells, and then the red blood cells, the packed red blood cells below that, and that's called hematocrit. 45% cellular component, 55%. Those numbers I want you to remember. Okay? 45% cellular component, what we call the formed elements are the cellular components, and 55% plasma, with a fair amount of variation. So it's not impossible for your hematocrit to be more than 50%. Males have slightly higher hematocrit than do females, about 5% higher, just owing to the greater rate of hematopoiesis that males do. All right. This is stuff we'll talk about in lectures, so I'm not going to bore you with these details right now. What we want to get to is what are the cells that we find in here? The so-called formed elements. White blood cells, red blood cells, and platelets. And white blood cells are called leukocytes. All these things that you see in here, oh, not all of them, the ones that have these purple staining nuclei, those are all white blood cells. And again, the collective term for all white blood cells are called leukocytes, which we've got 5 types of leukocytes. The ones you see in the background here, those are your red blood cells, and they're called erythrocytes. Red blood cells are erythrocytes, white blood cells are leukocytes. Fragments of cells that are involved in clotting are called platelets, or they're called thrombocytes. That's what these are up here. These are thrombocytes. Thrombocytes aren't even whole cells, they're just fragments of cells. So your erythrocyte, red blood cell, has a diameter of right around 10 micrometers. This is biconcave cell. We've got some models over here that you're going to identify these. For lab, there are three basic things I want you to know about the cells. Got to be able to identify them, either as a model or on the microscope. You need to know their prevalence. That's how many per microliter you'd expect to see in a blood sample. Okay? That's called the prevalence. The prevalence of students in this classroom right now is 5 per classroom. Get the idea? Okay. The prevalence of red blood cells, I put this on the handout I gave you today. Did you get it? So what I have, 4.6 or 6.8 million, something like that?
4.2 to 6.
4.2 to 6.8 million. And it says per MCL, that's microliter. Okay. Everybody have one of these? So a microliter is 1/1,000,000th of a liter, right? A milliliter is a 1,000th, a microliter is a thousandth of that. So I tell students to visualize this, think of taking a teeny tiny little pin, and the smallest little drop you can get dangling off of the tip of that pin. That's about 5 to 10 microliters. Okay? That's a really small amount. So in that super teeny tiny drop, you'd have 10 times what's listed here. Okay? Without a micropipetter, you can't pipette things like 1 or 2 microliters. Now I worked in a molecular biology lab. We did this thing all the time, and you have these $10,000 micropipettes, and you work with enzymes that cost $500 per microliter. So, you know, you're kind of careful with what you do with these things. But that's not an amount that we're used to visualizing. So it's a super teeny tiny drop. And I'm only emphasizing that so you understand this is a very small amount. And in there you have millions of red blood cells. So you have trillions upon trillions of red blood cells in your whole cardiovascular system. Okay? All right, so I do want you to know that number. The number is not fixed in stone. Okay? You'll read in some books it'll say 8 to 9 million. Other books will say 3 to 6 million. It just depends on kind of whose numbers you're going with. So if you told me somewhere between 6 and 7 million red blood cells per microliter, good enough. But don't screw up the unit. Okay? Don't say per milliliter or per deciliter or per liter, because that's completely wrong. Okay? You got to have it with the right unit. Now look at the white blood cells in that same tiny little drop. You only have about 4.5 to 11.5 thousand in that same drop. So 1,000 is 1/1000th of a million, right? So we're talking about at least 10 to the third, 10 to the fourth less than the red blood cells. So that means that for every white blood cell we see in a peripheral blood smear, for every one of these, we have at least 100, but probably more like 1,000 of those red blood cells. They should outnumber them about 1,000 to 1. So here, this is actually a pretty high concentration of white blood cells per red blood cell. They got them all crowded together. Probably this is an acute infection that's going on here. All right, and then in that same tiny little drop, how many platelets do you have? Some books will say spot on 250,000. Here they give you a range, 150 to 450,000. These are normal numbers, so these are homeostatic set points, right? You go get a blood draw, you get this checked. If you have too many white blood cells, too few white blood cells, there are names for those things. So for instance, if you have anemia, you don't have enough blood cells. Okay? If you have polycythemia, you have too many blood cells. Okay? If you have thrombocytopenia, thrombocytes are the platelets, that means not enough platelets. Okay? Leukopenia, not enough white blood cells. Leukocytosis, too many white blood cells. So there are conditions associated with all of these issues. Okay? All right. So then you go down to the white blood cell. There are 5 types of white blood cells, and you're going to learn each one of these 5 types. So 3 things I want you to know about all the formed elements. First of all, how to identify them both on the models and in the slides. 2, their prevalence. And 3, just their rudimentary function. So red blood cells, they carry oxygen to the cells. That's their main function. So we'll talk about the white blood cells. You can arrange the white blood cells into groups. We call them granulocytes and agranulocytes. granulocytes are so called because they have these conspicuous dark staining granules. You've got three types of granulocytes based on the type of stain they take up. If they take up a basic stain, like hematoxylin, then they're called basophils. These guys. And that's a very dark staining type material. It's called a basophil. If they take up a acidic stain, they're called acidophils or eosinophils. That's these guys. They have red granules. Okay. Eosinophils, bright red. Basophils, dark staining granules. If their granules aren't highly acidic or basic, they're more or less neutral, and they're called
Neutrophils.
Okay? And they have kind of an intermediate staining type granule in them. Those are your three granulocytes: neutrophils, basophils, eosinophils. Okay? The other two white blood cells are the biggest ones, which are phagocytes, things that go around and ingest other cells. And they're called monocytes when they're not active, and when they're active, they're called macrophages. Okay? Before they're activated, they're called monocytes, and after they're activated, they're called macrophages. And these are agranulocytes, meaning they don't have conspicuous cytoplasmic granules. Much smaller ones, with a very round nucleus, these are lymphocytes. These are also agranulocytes. Lymphocytes are the main functionaries of your adaptive immune system. So we've heard more about immunity in the last 2 years than most of you have ever wanted to hear in your entire lives. These cells are at the heart of all the controversy about vaccines and natural immunity and blah, 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 blah. So lymphocytes are the ones that are involved in adaptive immunity. You've got two major types of lymphocytes. They're called B lymphocytes and T lymphocytes. Okay? Antibodies, that protein that we've heard so much about lately, they're produced by B lymphocytes. T lymphocytes, they're involved in attacking infected cells and anti-cancer, stuff like that. But we're not going to get into identifying the differences between T and B lymphocytes. But just know there are two categories. Okay? So lymphocytes and monocytes are your agranulocytes. It's a little more useful for a differential blood smear, that's what you see up on the screen there, to know the relative prevalences of all three of these. So that's how I'm going to go through. I'm going to go through them not in grouping them by granulocyte or agranulocyte, but by how common they are when you find them. Okay? Have you ever heard of polys? If you ever worked in a healthcare setting, people say, "What's their poly count?" Have you ever heard that phrase? No? Okay. Well, now you have. A very common site of acute infection is somebody will say their poly count is very high. And that is talking about white blood cells, but it's talking about a specific type of white blood cell. So you look up on there and you can see where it says neutrophils and it has two arrows pointing up there and it has those purplish red globs. Lower left, it looks like what you got, like kind of two small ones and a bigger one that looks like a figure eight. And then the one to the upper right is kind of like an S and another little dot. Those are the nuclei. So that is a nucleus and that's a nucleus. Those two nuclei look quite different, don't they? They're highly lobed. You're going to have, say, 2 to maybe half a dozen or so lobes to this nuclei. It's actually a single nucleus and it has these large lobes and they're connected by thin little strands of nucleoplasm. Since the nuclei look so different from one cell to the next to the next to the next within this group, they're called polymorphonuclear, which just means differently shaped nuclei. Leukocytes. Polymorphonuclear leukocytes. PMNLs. Okay? Since nobody likes to say polymorphonuclear leukocytes, they just call them polys. Their standard name is a neutrophil. Very rarely in laboratory settings do people actually call them neutrophils. Okay? They call them polymorphonuclear leukocytes, or PMNLs, short poly. Okay? These are your body's first responders to infection. They are short-lived phagocytes. Short-lived phagocytes. Which means they're roving around in your blood until they're activated. When they respond to chemical signals released by cells, usually called lymphokines, they become active, and they go and attack whatever the foreign material is if they can identify it. And the way they attack it is two things. They release their granule contents, which will contain antimicrobial compounds called defensins, and also an oxidative burst. Enzymes that'll produce superoxide ions that'll damage tissues, and that'll destroy invading living microbes. It can even inactivate some viruses. They will also phagocytize the particles. They'll take them in through endocytosis and pump them full of destructive enzymes to try to destroy them and inactivate them. Now, when they do that, they die. So neutrophils, once they become activated and they do their thing, they die. When they collect at a site of inflammation, that's what creates pus. The pus that collects is basically damaged cellular tissue and a whole lot of neutrophils. So that, when you have an acute infection, the numbers of these skyrocket. So the third thing I want you to know about your white blood cells is the relative prevalence of each one. And that's the percentage that I give you in your sheet. So you see it says neutrophils, 40 to 60% of WBCs. So that means roughly half of the white blood cells you identify when you look at a slide like this should be neutrophils. So when you look at a slide, you start seeing neutrophils in there, or we start seeing white blood cells. In your mind, the first thing you should think is, okay, most of these should be neutrophils, because that's what this number tells you. So you can take a white blood cell count of 10,000, and what you can say is, well, roughly 5,000 of those, 4,000 to 6,000, should be neutrophils. So when you do a differential blood smear and a count in a laboratory in a hospital, and you get back the numbers, and it says, okay, we're seeing 18,000, a PMNL count of 18,000. Right away, that tells the clinician that that's an acute infection. This person is suffering from some sort of acute infection. We don't know what it is, but they're clearly having a large-scale immune reaction because their neutrophil numbers are through the roof. And the only reason your body does that is if it's actively fighting an infection. So that's one of the useful things that can tell you. Okay. So, first responders, short-lived phagocytes. Got that? Okay. Moving on to the next most common white blood cell. So this one is actually an agranulocyte. This is a lymphocyte. So neutrophils are granulocytes. They're the most common white blood cell. And then you move to the lymphocytes. They're the second most common white blood cell. And I told you what these do already. The lymphocytes that you find in circulation are mostly T lymphocytes. They're almost all T lymphocytes. And T lymphocytes are part of your adaptive immune system, primarily. And they mediate something we call your cellular immunity. In order for a virus to cause infection in you, do you know what the virus has to do? All viruses, this is true. This is something that all
Multiply.
viruses do. What's that?
Multiply.
Yeah, and how do they do that?
Go to use the cells, ribosomes.
Okay, so they have to get taken into your own body cells. They need a host cell. Okay? Viruses can't do anything unless they get internalized by your cells. So cytotoxic cells, which are activated T cells, one of the activated T cell types, their job is to go around and actually destroy virally infected cells. And they just kind of short circuit that replication cycle. So a virus gets into a cell, the cell sends out little flags on its membrane that say, hey, I'm infected. And T cells will come by and they'll kind of check and they'll say, yeah, you are infected. Sorry. And it'll inject them full of chemicals that cause its cell to die. In so doing, it interrupts the replication cycle of the virus. That's how it protects you. It'll do that same reaction with cancerous cells. So when these T cells recognize abnormal body cells that could become cancerous, it tags them for destruction. So we have white blood cells that are designed to protect us from viral infection and cancer all the time. It's not a perfect system. Obviously, sometimes we do get infected and sometimes we do get cancer, but it's a pretty good system. So those are your T lymphocytes, and that's what this little guy is right here. The B lymphocytes, they're responsible for what's called humoral immunity, and this is the one that produces antibodies. The B lymphocytes most of the time stay in your lymphatic organs. They stay in your lymph nodes, so they don't really migrate out into the blood. So when we isolate those blood smears, it's going to be T lymphocytes, not B lymphocytes that we see. And we'll leave it at that for now. Upwards of 20% of your white blood cells can be lymphocytes. What did I give you on here? Yeah, 20 to 40%. 40% would be way on the high end there. So 20 to 40% will be our range we'll learn for lymphocytes. So now if you think about this, if you've got 50 to 60% neutrophils, 20 to 30% lymphocytes, we're dealing with somewhere between 80 and 90% of our white blood cells. So when you look at a smear like this, it should be neutrophil, 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 lymphocyte, neutrophil, neutrophil, lymphocyte, neutrophil, that's what you should see. The three remaining classes are fairly rare. The next most common type of lymphocyte is a monocyte. Somewhere between 6 and 8%, 2 and 8% is what I put on the sheet, 2 and 8%. And this guy over here is a monocyte.
You said
Monocyte.
that was uncommon? I'm sorry.
Say it again.
What did you say, it was the most uncommon?
Well, the three remaining types are far more uncommon.
Okay,
So your two most common types are neutrophils and lymphocytes.
right,
And the three remaining types are fairly uncommon.
okay.
The third most prevalent would be the monocyte. Remember, when it's inactive, it's called a monocyte. When it becomes active, it enlarges and crawls out into the peripheral tissues, and there it's called a macrophage. Macrophages do a couple really important things. One, they're long-lived phagocytes. So these things will ingest foreign material, just like a neutrophil, and try to destroy it, but they don't die. Macrophages don't die when they do that. Instead, they chop up that foreign material and they put it on a cell surface marker, which then they take and show the B lymphocytes. And the B lymphocytes will recognize that foreign material and they'll start producing antibodies in response to whatever they're shown by the macrophage. So macrophages are roving around out there in the dangerous neighborhoods and looking for bad things, and when they find them, they internalize them, chop them up, and they're like, okay, it's kind of like taking that photo back and showing it to the, the authorities and saying, hey, look, I saw these bad dudes, go get them, okay? So now they know what to find. And so the lymphocytes start cranking out antibodies, and those are the things that go out and fight the dangerous materials. We'll talk in lecture when we do immunity, we'll talk about how this all works. I'm not going to get into that here. Suffice it to say, if you're a law enforcement individual, TSA screener, whatever, and you know who you're looking for, you can do your job a lot better, right? When macrophages are looking for foreign material that have been tagged by antibodies, their ability to clear the system from that goes up 10,000-fold, okay? Which means instead of clearing out one virus particle per second, they clear out 10,000 virus particles per second. Much more efficient. So macrophages continue to be very, very important in all of your immunity, but they can become a whole lot more active and a whole lot more efficient when coupled with the action of antibodies. Okay? All right. So that's one thing. They're they're long-lived phagocytes. They internalize the material, they chop it up. That process of showing that material to the B lymphocytes is called antigen presentation, if you want a name for it. It's called antigen presentation. They're one of three types of antigen-presenting cells in your immune system. They also secrete a lot of things that are called cytokines. Cytokines are chemicals that other immune cells respond to. Have we ever been at a family gathering or something like that, and everybody knows when dinner's ready because you can smell it? And everybody starts migrating towards the kitchen because you're all smelling this wonderful smelling stuff. All right. That's what we would call positive chemotaxis. You're moving towards the source of that wonderful smell because you want to know what it is. Okay? Macrophages, when they encounter foreign material out in the periphery, they secrete chemicals along with the infected tissue that act that way. Those chemicals are called cytokines, and other immune cells go flocking to that area. What's your first responder immune cell?
Neutrophil.
Neutrophils. How do you suppose they know where to go? This is how, okay? They respond to cytokines that are released by the damaged tissues and the macrophages, and then those neutrophils flock to that area. It's called positive chemotaxis. That's another job that macrophages will take care of. Okay, and then that leaves us with two types of granulocytes. In order of their prevalence, you would have eosinophils, followed by basophils. Eosinophils are going to be like what? 1 to 2%? Cheat here. 2 to 4%. 2 to 4%. And then basophils a little less than 1%, about 0.5%. Eosinophils are the bright red staining ones. The basophils are the ones that stain a very deep bluish black color. Eosin is that, it means red. Eo means red. So these are red staining cells. Well, actually I have a picture of it right here. But we do eventually. All right, so neutrophil, that's an eosinophil. See how red the granules are? All right, true or false? Staining techniques never vary.
False.
Very false, right? Okay, we've looked at enough histology slides that you know from slide to slide to slide, you're going to see some pretty serious variation in color, right? So when you look in a blood smear, what students always they want it to be simple, right? You want to look at neutrophils and go, okay, those are kind of intermediate staining, basophils are dark blue, and eosinophils are bright red, and there's never any ambiguity, which of course is nonsense. So when you look in these microscopes, you have to compare. You have to look at the red blood cells and how intense that red staining is. And you look at the neutrophils. Are those granules red, purple, or blue in that neutrophil?
In the red.
I got some votes for red. How about that one?
Blue.
Wow.
Purple. I don't know.
Exactly, okay? You can make a case for any of those three answers in this picture. Depending on who stained this and how well they stained it, those granules and neutrophils might look a little blue, they might look very red, or like this one, kind of a combination in between, which is really what you'd expect because they're neutrophils. But there's no mistaking this for neutrophil because of that highly multi-lobe nucleus. Only neutrophils will ever look at that. So I always tell students, use that as your baseline. Identify a neutrophil, kind of get a feel for how those granules look. I'm telling you this because if these granules look slightly red, you're going to feel like you see these things everywhere. Okay? Because the granules in most of the granulocytes are going to look a little reddish. But if they have a highly lobed nucleus with multiple lobes, they're neutrophils. If you start thinking you're seeing hundreds of eosinophils, you're not. Okay? Those are neutrophils. When you look at an eosinophil, the granules in the eosinophil should be like neon red compared to the granules that you see in a neutrophil. If, on the other hand, you look in the neutrophil and those granules look kind of pale blue, then the chances are that the granules in this eosinophil are not going to be bright red. They're going to be kind of a pale pink. But compared to the neutrophil, they are going to be several shades, if that's a thing, several shades brighter red than the neutrophil. You only get good at this by doing it, right? You look at a perfect picture in a book, and then you look at that on the slide, and well, that's not the reality. You have to compare for yourself a few times. So we're going to have a few chances to look at these. I will say this, when I put an eosinophil on the test, I only picked one that has pretty conspicuously red granules. So I'm not going to pick one that's kind of close.
In between.
So what are eosinophils? So 2 to 4% of your white blood cells. They have two major functions I want you to be aware of. Okay? Eosinophils are anti-helminthic. In other words, they fight roundworms. Helminthic.
How do you spell that?
How do I spell it, or how is it spelled?
Both.
Helminthic. Helminths are small parasitic worms. Okay? Which is kind of gross when you think about it. Okay? That we have a white blood cell type that seems to be dedicated towards fighting parasitic worms. In the United States, we don't get parasitic worm infection very often. I was a dirty, grubby kid. I played in the sandbox all the time. So I got roundworm infections when I was a kid because the eggs are everywhere in the soil, and so you play in there and you pick your nose or whatever, and you get this. It's very common in kids. That's about the only common roundworm infection you get in the United States. Not true in the rest of the world. Okay, in the rest of the world where we don't have really great sanitation, you get roundworm infection all over the place. Okay, filariasis, enterobius infections, Shigella infections, Ascaris infections. So it's very, very common in Southeast Asia, the Brazil, Amazon basin, lots of places. This is very, very common.
Are like tapeworms considered like element?
Yeah, tapeworms are flatworms too. Yep. These are most effective against the roundworms, but they are somewhat effective against tapeworms and other types of infectious worms like flus and stuff like that. So it tells us from an evolutionary perspective that these types of parasites have been problems for vertebrates since like the beginning of time, and that we have a red blood cell dedicated to fighting those types of infections. So that's function number one I want you to know. And function number two, they secrete a compound called histaminase. Histaminase.
I was just about to ask you to spell it.
Now you're probably familiar with histamine, but that's not what this is. This is histaminase. This is an enzyme that breaks down histamine. So this is a natural antihistamine. Histamine is a chemical that promotes inflammation. To shut off inflammation, you need another chemical, and that's this. So eosinophils are important for shutting down inflammatory responses. Okay. Last time, and before I leave this, I'll just mention this. You don't really have to remember this, but it's an interesting little factoid. Some of our autoimmune diseases, like irritable bowel syndrome and Crohn's disease, there's a genetic component to those, but we know in those disorders that it's out of control inflammation in the bowel that seems to be causing the problem. And for whatever reason, eosinophils seem to be implicated in causing some of that inflammation in the bowel, as if they're attacking parasitic worms, but they're not. And it's an inappropriate inflammatory response. It's an autoimmune disease. It's been shown that you can actually treat some of these disorders by infecting the person with roundworms. Okay, so they actually give them what are called benign roundworm eggs, and so like I was talking about the enterobius infection, pinworm, these tiny little white worms that we get in America. They don't really cause much problem, a little bit of discomfort, and that's it. Those types of worm eggs will give the eosinophils something to do, okay? And then they wind up not causing out of control inflammation. It's actually been very, very successful in treating and controlling things like IBS and Crohn's disease. Since this was first discovered, of course, companies have been trying to figure out what the chemical mediators are in these eggs that are causing this, and they're trying to encapsulate that and turn it into a medication with some success. So now you have some medications that are aimed at sort of giving eosinophils something to do so they don't cause the irritation. It's one of those situations where really good sanitation has mostly removed things like parasitic worm infections in the United States, and so now we wind up getting an autoimmune disease as a result. It's pretty tightly tethered to the concept of the clean hypothesis, which we talked about a little bit in micro. It really is kind of the damned if you do, damned if you don't sort of phenomenon. It seems like our immune systems work best when they're challenged by things. But the flip side is, if you never challenge it, your immune system gets a little bored and starts attacking things that maybe it shouldn't otherwise, and that's involved in things like lupus and rheumatoid arthritis and all sorts of autoimmune conditions. So there's probably a happy medium in there where you challenge your immune system on a regular basis, but of course you don't want to challenge your immune system with Ebola. Okay? That's maybe not the best way to go. Yeah, so we'll leave it at that. I don't want to spend any more time on that right now. Granulocytes, the least common white blood cell. So less than 0.5% of your white blood cells are granulocytes. I didn't mean granulocyte. Basophil, sorry, basophils. They are granulocytes. I just read the top of the slide and shut my brain off. These are basophils. Okay. 0.5% of your white blood cells, very dark staining granules, fairly big and lumpy. The two main things that basophils are responsible for, they promote inflammation. by secreting histamine. Histamine is the most common cytokine.
Okay, so quick question. So, because these are for like the when people are allergic to like cats and dogs and stuff, right?
Yeah.
So if they're the least common, then why are so many people like? I don't want to
So
say
in
they're.
allergies, it's usually not the basophils, it's
Okay.
usually a connective tissue cell called a mast cell that does the same thing. But you're on the right track.
Okay.
To answer the question, why? Nobody knows. Nobody knows why we have allergic reactions. What we
Okay.
can say is that allergic reactions are inappropriate immune reactions. So why does bee venom, why does cat dander, why does peanut dust evoke such a strong immune reaction? We don't know. There's no good reason for it. Problem
Would
is,
that increase those numbers though?
oh yeah,
So then why are they considered like one of the least? Because I feel like everybody I know has an allergic
because when you're
reaction.
not having an acute inflammatory reaction, the numbers are very low.
Okay.
Right? So you take a peripheral blood smear from any of us in here right now that aren't having eyes watering and puffy noses and all of that, you would find very few basophils. Right?
Okay.
But if you're having an episode of hay fever, then the basophil numbers are going to be up for sure. It's not going to happen instantly. So if you're like chronically exposed, if you're allergic to cats and you're chronically exposed to cat dander, yeah, your basophil numbers are going to be triple, quadruple what they ordinarily would be.
Okay.
And in fact, when they take a blood smear and they look at that, they're going to see that both your basophil and your eosinophil numbers are going to be quite elevated. And they're going to say, have you been sick lately? And you're going to say, no. And they're going to say, do you suffer from allergies? And you say, oh, yeah. Okay. And they're going to know that.
Okay.
All right. So these granules here contain histamine, which is the primary pro-inflammatory cytokine. And also they contain a chemical called heparin. Okay? And heparin helps break down clots, which is important for an immune reaction because you need to get your immune cells into the damaged tissue. So basophils show up, they secrete a clot-busting chemical that helps other white blood cells get through that clot and get into the infected tissue or damaged tissue to help repair it. So two main things you find in the granules of basophils, histamine and heparin. Okay, we good with that? All right, so I'm going to stop here. You can have the rest of the time. What I want you to do for sure today is just maybe take a picture of the model.